So hi everyone and welcome to RIOTS. To all of our regulars, welcome back. If this is your first time, RIOTS is a self-organizing initiative that is entirely ECR-led. We host weekly seminars that aim to raise awareness and provide training in reproducible, interpretable, open and transparent science. Do please follow us on our social media, Twitter, YouTube, etc., and engage with our work. So we are almost at the end of the term, but there are still a few events that you can join before the summer break. So right after this talk, you can join the mini conference on teaching and mentoring open research. And tomorrow, you can also join an awesome talk that is organized by RIOTS Rotterdam on Indigenous knowledge systems and epistemic pluralism. So before we introduce our guest speaker for today, just to do a bit of housekeeping, as you can tell, we host our events on MS Team Live Events. This generates a publicly accessible link so that you can share this with anyone and you can share this on your social media as well. It is a one-way streaming platform, so you can ask questions via the Q&A message board. These will be moderated by myself and my co-producer. Do keep the questions and comments obviously relevant and respectful. We'll feed them to the speaker at the end of the talk. But just in case we have many questions, do make sure that you upvote any questions that you want us to prioritize, and we'll make sure to address them in that order. If you joined late, also don't worry, you can pause, rewind, and fast forward. And live events also has a really good auto subtitling feature too. So if you need that, you can turn that on as well. So now on to our main event. So today we're joined by Eilidh MacNicol, who will be talking about species-agnostic tools for translational MRI processing. Eilidh studied neuroscience at the University of Glasgow before coming to study neuroimaging at King's College London. Her PhD research used MRI to investigate how brain networks change in healthy aging. And during this time, she became interested in extending processing tools that were developed for human data to be species agnostic and supporting open science and reproducibility in the preclinical MRI community. In her current role as a postdoctoral researcher, she continues to apply network-based analysis methods to MRI data with the aim of modeling brain changes over time. You can find more about Eilidh on Twitter and on her website. So that's enough from me. I'm going to let Eilidh start now. So good luck, Eilidh. We're looking forward to hearing your talk now.
Thank you very much for having me. Let me just get my slides. Okie doke. If you could just confirm that you can see that okay?
Yep, all good. Yep.
Great. Well, yeah, thank you very much for having me speak today. I'm Eilidh, as I just introduced. Today, I hope to convince you that most of the MRI processing software that we use assumes human adult data as the norm, and that this is a problem for reproducibility because it causes unnecessary divergence in an otherwise very translational method. So I'm going to suggest some solutions, but ultimately this can only be solved by the neuroimaging community with its wide range of applications and its diverse backgrounds coming together and collaborating on a solution. So first, some context. This was me. I was at the top of Mount Stupid. I did a master's project where I fed my human data into my chosen neuroimaging software with all of the defaults. It went from nose to tail, and I got acceptable quality results at the other end. So when it came to my PhD, I thought I knew what I was doing when it came to neuroimaging analysis, but I don't think I really understood the assumptions that underlie the individual processing steps. It was very easy for me to, go through the motions and get what I needed at the other side. So I thought I could focus on my PhD project, and instead I spent a lot of the next 4 years trying to optimize the processing pipelines I was working on. So a lot of this work today has been motivated by the time, because I don't think anyone else wants to make the fast descent down Mount Stupid like I did. So yeah, here we go. Our outline for today. So first I'll give some context to what preclinical imaging is, because I don't expect everyone to know it. And why it's different from normal quote unquote imaging of adult humans. and how these differences feed into the problem that causes a divergence in the field. I'll go on to discuss NiReports, which is the neuroimaging pre-processing tools ecosystem, and I believe that this particular ecosystem provides a flexibility to facilitate a bidirectional translation, both from human adults, to other populations, but also from these other populations to give us information for human neuroimaging. Finally, I'll talk about a few of the tools that we've taken to extend into rodents in particular, but with the scope to extend into other species and populations with future collaborative efforts. So first up, yeah, let's address what I mean by preclinical imaging, because I'm going to use that term a lot. I'll talk mostly about my experience of imaging with small rodents like rats and mice, because that has been my experience. However, there are way more different types of animal imaging, including other animal models or MRI for veterinary research. But when I say preclinical imaging, I essentially mean preclinical imaging of non-human MRI. That is to say non-clinical MRI. So here's my hitchhiker's guide, so don't panic. The first thing most people ask me about scanning small animals is, do you have a small MRI scanner? Well, the answer is no. We just have one with a tiny bore. So this MRI is an ideal translational technique because it's theoretically analogous across species, because essentially the same physics is happening in the same across all of the species. It's also non-invasive, which appeals to a central tenet of preclinical imaging, which is to reduce the number of animals used and to refine the harm that we cause to them. For those of you who are not sure of your rat anatomy, this is a T2 weighted anatomical scan of a rat, and we have the brainstem here, and we have the cerebellum. We have the corpus callosum, is this dark stripe here. This is the dorsal hippocampus, anterior commissure, the eye, and the olfactory bulb. This isn't a test, it's just in case you're looking at some of these images later on to make sure that you're orienting yourself so you know where you are in the brain. So why do we do preclinical imaging? Well, preclinical imaging connects basic science and human medicine, that is bench science, all the way to pharmaceuticals or interventions that allow us to help people's lives be better. And the key part about preclinical imaging is that it allows us to combine techniques in the same subject that we might not be able to do otherwise. So, we can do our basic science techniques, our wet lab techniques in cells, but also in animal tissue. And while we can do them in humans, it's maybe a little bit limited in terms of how easily we can get those samples, or we might be time restrained. So it maybe compromises our results. But on the other hand, we can't really ask cells to do behavioral tasks like we can in humans, but we can also do that in animal models. So only in animal models can we actually, link these two facets of neuroscience together. But unfortunately, there is a downside, and that is that they're not exactly the same for a couple of different reasons. And I feel like they boil down into one of these three categories. That is anatomical differences, the study design, and the software design specifically for neuroimaging. And I hope that this talk will convince you that the first two may be difficult to change and to overcome, although we are trying. But hopefully I can convince you that we can improve the last one, and any differences don't need to be there. So the most obvious first difference in anatomy is the size of the brain. So the human brain here with an average of about 1,400 grams compared to the rat, which is about 700 times smaller than the human brain, and the mouse brain is smaller still. This is obviously going to take into account a lot of the size of the images that we're acquiring. This is probably the most obvious difference. The next difference that is quite obvious is the difference in ratio between grey matter, white matter, and CSF, cerebrospinal fluid. That's because there is less grey matter folding. So there is a different ratio to grey matter to white matter, and also this cortex to subcortical ratio is different because humans have a lot more cortex compared to rodents. The other obvious difference is the olfactory bulb, which is much bigger in rodents than it is in humans. But although there are these large anatomical differences, many of these can actually be overcome by using resources that are species specific. So maybe that's templates or tissue probability maps or atlases that are specific to the animal that you're using. But as a consequence, we need to have some software that that can take these different resources with us. But I'll talk about that some more later. The other thing that we have to abide by is the 3 R's of animal research, which greatly impacts our study design. So if we can't replace animals in our studies, then we have to reduce the sample sizes and refine the treatment to ensure that minimal harm is done. So consequently, we have smaller sample sizes, but this can somewhat be compensated by the fact that the brains of the rodents are more similar to each other than a human to each other. That's because there's less cortical folding, as we already discussed. There's also, as we've talked about, non-invasive. MRI is non-invasive, so we can, repeat our studies within the same subject and reduce the variability there too, which helps to compensate for some of the small sample size. However, for all that we can kind of overcome these small sample sizes, there are fewer large-scale sharing initiatives. There are a number of smaller multi-site studies that are becoming more popular, but we really don't have this sort of scale that the UK Biobank, Midnight Scan Club, My Connectome, Human Connectome project has been able to offer. And this might be specific to animals because although other human populations do have some efforts, including like the Developing Human Connectome project and the Baby Connectome project. So yeah, these are specific to small animals, it seems. The other thing we have to account for is the animal's physiology. Now, it's really hard to get an animal to stay still in the scanner, so we use anesthesia a lot, but this unfortunately has an effect on brain function and the neurovascular coupling, which we kind of rely on for our measurements when it comes to functional imaging. So you can see here that these are, you can see that this is the connectivity matrix for when an animal is awake and the effect of several different anesthetics have on the correlations between different brain regions. And you can see, for instance, isoflurane has quite a lot of big changes from awake, whereas, we've managed to, you can also see this in the right-hand side, which is an EEG trace under different anesthetic protocols and how different single cells or single regions are reacting to anesthetics. Fortunately, the field is kind of going in one of two different ways, in the sense that awake imaging is becoming more popular. There are more and more paradigms becoming available for people to try, but also there seems to be a consensus emerging in the field with respect to choice of anesthetic regimes. So a couple of these big multi-site studies have got together and have seemed to agree on the best way forward that replicates awake scanning as best we can while still under the effect of anesthesia. One of the other big differences is the actual acquisition in our study design. So there is a much more different landscape in terms of scanner vendors. So in human scanning, there are maybe 3 or 4 different big name vendors, whereas in preclinical imaging at the moment, there's really only one. So that also influences the coils that we use to actually acquire the image. So we are much more likely to have maybe homemade coils, particularly if someone has a scanner that's no longer supported by the manufacturer. And there may be conversations to be had about whether the purchase coils versus the homemade coils are more reliable for quality of images, because homemade coils are maybe more likely to be tested on a daily basis compared to purchase coils, which may get a service, you know, every 6 months, 1 year, or when something goes wrong. So yeah, then we also have decreased head motion compared to human acquisitions, because the animals are usually, apart from being anesthetized, are usually restrained in some way, maybe with bite bars or ear bars or a stereotactic frame. But this also does mean that head motion is much less of a concern, generally speaking, than in human MRI, where the participant is much more freely moving. There's also a lot to be said about the actual imaging protocols and how different they can be. So generally, the anatomical scans in humans are T1 weighted. That's because they provide really good contrast between the grey matter and white matter images, but T2 weighted images are much more commonplace in rodent imaging. So that's partially because of the high field strength, but also because of the lack of need for this contrast because of the difference in grey matter and white matter ratios. But even if you did have an equivalent sequence, there's also an issue about whether the task that you're using is also an equivalent. So even if you could set up a completely homologous sequence, would the task actually reflect what you're saying you're trying to emulate? For instance, we can't do, if we have anesthetized animals, we can't do behavior in the scanner the same way you might be able to do a cognitive test in the scanner. Also, the image field of view has to change because of the anatomy. So brains are generally longer from the brainstem to the olfactory bulb, whereas human brains are much more spherical. So to overcome this ellipsoid versus sphere problem, we either need to have more slices, which increases the image size, or we need to have anisotropic voxels in the sense that one dimension is much chunkier than the other two. Or we need to have a smaller field of view, in which case, you know, we only have like a single slice. And in that slice, there's also a difference in the proportion of the background. So in rodents, there's much more of the skull and the muscle that might be much thinner, much less compared to air in humans. And finally, there's also a much wider range of scanner strengths than available in humans. So most, I would say, human work is being done currently at 1.5 or 3 Tesla, but obviously with exceptions for the breakthroughs in low and high field respectively. But in preclinical imaging, we have a much wider range from 4.7 Tesla all the way up to 15 and sometimes even higher. But the important thing to note here is that the spatial resolution is not proportionate to the field strength. So just because, so we talked about earlier the rat brain being 700 times smaller than the human brain. Well, we're not using a scanner that's 700 times stronger. So our spatial resolution does have to be a compromise on that respect. So essentially, the things that I've discussed already is that preclinical workflows lack unification. That's partially to do with the study design having to be different and having to design a study towards your own research needs, but also when it comes to the actual processing, because as it happens, most of the software is developed for human data as an assumption. So what researchers who use preclinical data might do is they'll come up with a way to make their data work with the existing software, or they'll write their own software, or basically they'll have to change the software from the one that's used for humans for one that's made for rodents. And this provides a divergence between species. So the human studying population is going one way and the rodent studying population is going another way. But this also causes a difference between groups, because essentially the groups are doing slightly different things to overcome the software between themselves, which makes it really hard to compare between sites sometimes. This has some serious implications for reproducibility, because how do we really know that one site is doing the exact same pipeline as another? But it also puts up a barrier for information flow between fields. Some of this work was inspired by the fact that researchers had come to us because they had in fact they'd put a call out on Twitter, and they didn't know how to handle the rodent data that they'd been given by a collaborator, even though they were very experienced in human imaging. So yeah, I suppose that this barrier is actually stopping people from it's making people realize that that's where I found myself after falling from Mount Stupid. It's not to say that it's like completely a wasteland. Basically, there have been a couple of tools that have come out where they've tried to do a similar pipeline that is available, but specifically for small animals or for experimental animals. But the problem for most of these is that they are nose to tail, much like the other human analysis, in the sense that they are, you put your data in at one end and you get your results out at the other, and you're not really sure what's happening in between. Plus, these tools are effectively reinventing the wheel, but with rodent data instead of human data. And so we're still not really getting to the nub of the problem, which is that we're still diverging. We're just kind of doing it synchronously in a particular species. So there's no real consensus yet, but I hope that what I'm going to go on to show you is that with some collaborative efforts, hopefully we can actually reach that. So I'm going to talk about the NiReports tools now, which is neuroimaging pre-processing tools that are specifically geared towards openness, transparency, and also specifically only to the pre-processing part of the workflow. That means that as far as the outputs are concerned, they are completely agnostic to any of the analysis techniques you might want to do. So there are no assumptions made on the part of the software for whatever kind of analysis you might want to do. For instance, if you were to do some sort of voxel-based morphometry where you might want some smoothing, you can apply that smoothing yourself. If you have to regress out nuisance factors, it gives you the nuisance factors, but it doesn't regress them for you. There are no assumptions on the actual outputs. It just gives you everything you need to make those decisions as a researcher yourself. So NiReports is an ecosystem of tools, and everything in the brain in this pyramid are actually maintained by the NiReports community. There are a handful of tools here in the bottom right corner, BIDS, NiBabble, and NiPipe, which are not actively developed by the NiReports community, but they are very much, central to it, and there's a lot of collaboration between the open source software communities in this respect. So NiReports aims to give data effectively the sashimi grade fish treatment. That is data that is minimally pre-processed but safe to consume directly by your statistical analysis. So it's separated on this pyramid from end user projects, which some people might be familiar with, and fMRIPrep or MRIQC, through to some middleware products where people are maybe less likely to interact with them themselves, down to the bottom, the underlying software infrastructure, which is mostly intended for developers. And one of the key things about this ecosystem is that it's supposed to be about minimally pre-processing, which also involves a minimal user intervention. So most of these tools are run from the command line with a single command. But is minimal user intervention actually desirable? Well, minimal user intervention means that you have less parameter tuning from the researcher. So say you think that the default setting didn't work, you go, okay, well, I'll just put that number up and see if it runs any better. Oh, it didn't work, I'll put it down, I'll see if it runs any better. So that might be a source of bias that we're not accounting for when we're actually recalling our results in the methods. It also has a lower barrier to entry because you don't need to know all these secrets that the preclinical imaging community have found about how to get their data to work with other software. You can just run it as it is. But the problem with the minimal user intervention is that, one size doesn't always fit all. That's why defaults are normally available to be changed, because not everyone's data is the same, and they're collected for different outputs, so they're asking different questions, and we shouldn't assume that all of our data is going to be the same. There's also fewer opportunities to learn through practice. So, I mean, if you take my descent down Mount Stupid, then what happened was, as soon as I put data in and it didn't meet that default assumption, I had to learn how to change the defaults. I had to understand what all of the individual steps mean. And also it affects the transparency in this way, because it's not entirely clear at what step what is happening and why it's important to the processing pipeline. So I'm going to focus on this half of the table to show you why NiReports, I think, provides more flexibility than other tools out there, and why this might be prime for allowing us to use our rodent data with NiReports. So the first tool that we use a lot of is the BIDS. So all of the NiReports tools are BIDS apps, and that's the Brain Imaging Data Standard. That is simply a way that has been devised of storing your imaging files and the associated metadata in a way that encourages sharing and transparency. So, and on your left, you have maybe like a traditional DICOM folder, which is, it will maybe give you like the date that the scanner was, the date of the scan session, and then maybe some arbitrary number, maybe some subject number. But there's no real information here other that a human could read and think, oh, that's, useful information. On the other hand, the BID standard on the right allows you to see that there's a top level participants file in a standard tab separated format, and each subject has their own folder, and within each folder the images are separated into anatomical or functional or diffusion scans, and each of the images also comes with a JSON sidecar, which is a type of text file that provides you with information about the file acquisition that can be used for pre-processing later on. So the other thing that NiReports heavily relies on that allows it to have this flexibility is template flow. So template flow is a repository, a cloud-based repository of brain templates. And we've been working on this quite a lot to make sure that it's not just human templates, although there are a lot, and mostly the M&I ones, but we also have rat templates, we have macaque templates, we have human baby templates, we have human infant templates, we have surface-based templates, and we have volumetric templates. All of these are stored together in a cloud-based repository, and if there is any update to them, say that someone adds a new set of ROIs, we can version control them the same way that you would with GitHub or other version control systems, that allows us to be in real control over which version of an atlas we're using for our analysis. So this is particularly useful for workflows because it programmatically allows you to access whichever template you want, and possibly to even include multiple templates in your analysis, maybe making way for some sort of multiverse analysis where you intentionally change and use this and run the same study several times but with different templates. So as far as the learning opportunities goes, the NiReports is a part of the NiReports ecosystem that all of the end user tools generate these HTML reports that if you scroll through it, it will give you both critical quality assessment information, but it will also help you to understand the workflow, because you'll be able to see all of the key points that has happened in the workflow and the outputs of that key stage. So if you can see that there are examples where there have been segmentation contours laid on top of the image. There are examples that flash between a template image and an image that was registered to it. There's a series of different metadata available, including, if I just skip through this a little bit, you can see here that these are some of the components that might go on to use as regressors in our functional connectivity analysis, potential sources of noise, and it makes it very easy for you to digest just how good the quality of your images are. And these reports are fundamentally underlying all of the NiReports end user reports. So in addition to that, there is also citation boilerplates that are released as CC0, so Creative Commons license boilerplates, and these are available in 3 different markdown, or 3 different versions, like an HTML, a markdown, and a LaTeX, and it counteracts black boxing of the workflow. So it is increasing the transparency of every single step and providing references and citations for all of the tools that you use, but also the versions of the tools that you use, so that you have a real understanding and a control over you know, what is in your methods, but also what is in your, nope, sorry, lost that, is in your methods. So I hope that so far I've given you an idea of there's some context, both in preclinical imaging, but also NiReports. If there are no questions, then we can go on to how we started to tie these 2 concepts together to create NiReports tools that are inclusive of preclinical imaging and other species as well. Okay.
Yeah, we'll take them at the end, Eilidh.
Okay, sure. Cool. So the first of these tools was atlas-based skull stripping tool called RIOTS BrainExtraction, and this is the GitHub here if you want to check it out. So from speaking to other preclinical researchers, it was pretty clear that a robust skull stripping was one of the steps that most people had the most trouble with. So skull stripping or brain extraction is the removal or masking of parts of the image that are not of interest. So particularly removing any tissues that are not the brain in our case, but it could be anything. So essentially we're just left with the tissue of interest, and that allows us to reduce the inference from tissues of non-interest. But most of the tools that exist in this respect are aimed very much for the image on the left and not for the image on the right. Now, these are both T1 weighted images of the brain. It's just that the left is one of a human brain and the right is one of a rat brain. You can see, as I discussed earlier, that there's a much bigger difference in the terms of the bone and the fat available in the image. There's much more bone and fat in the rodent image. But also the background. So I think that this human image has maybe been cropped a little bit, but the background is normally much more air compared to the rat image on the right, where there's only really the top part that is background air. So most of these tools that we use are assuming some sort of contrast or shape that's similar to the image on the left, but these assumptions don't really stand for images on the right. And people have made tools to overcome this, but they are normally dependent on the fact that it's a T2 weighted image, or the fact that if they do like a threshold above this certain point, then it will work, but this might not work for reduced field of view. And basically it seems that there was a lot of options out there, but none were really robust across many different data sets. So what we decided to do instead was use an atlas-based skull stripping, because in this way we're actually exploiting one of the things about preclinical imaging that is not true for humans, in that the shape of the rodent brain is much more similar between rodents than between humans. So atlas-based skull stripping is in its most mature implementation in the ants package, and specifically its tool ants BrainExtraction. So the concept of atlas-based skull stripping is just that we have a transformation of a subject image to a template space. And in this template space, there is the template image, but there's also an associated mask. So if we calculate the forward transform to this template space for the subject image, then we can quite simply use the inverse transform to mask the subject space. So that's all atlas-based skull stripping is. So we effectively optimized the ants algorithm and called it ARPS BrainExtraction for atlas-based rodent tools BrainExtraction. And there is some massaging of the image to help facilitate the image registration, but it's a very fast image registration where we're focusing less on the details within the brain, but more just on the image boundary, which is why the Laplacian filtered images in the bottom here are so important, because that allows us to really well define the edge of the brain itself. So going forward from this, we've managed to find that ARPS BrainExtraction has been very reliable in rodents, but there's also been some parallel work in the NiBabies tool, which is pre-processing for babies, human babies, and we're hoping that actually maybe we could put all this information together into one tool. So the next step forward, we hope, is that we can use NiRodents and NiBabies to become a more generalizable brain extraction. These atlas-based brain extraction tools aren't particularly novel, but what is novel is that with the inclusion of template flow, we can actually have this switching between whichever template we want. So we can have the same researcher use a baby template with the same command line, except for just changing out the template, or they can have a rodent and just have a rat-based template with the exact same tool. That is ideally what we'd have with absolutely minimal user intervention. So the next tool is MRIQC, which provides quality metrics for assessment and control of data quality. So just quickly as an aside, we recently ran a symposium on quality control and assessment at this year's ISMRM, and we've written this Jupyter book with executable code. It's available at the link seen down here. And we're looking, if people want to help us collaborate, all the code's on GitHub as well. But essentially, we ran this symposium because quality is incredibly important for images, particularly when we need to make assumptions about the underlying biology. We need to have accurate representation, and quality images helps us do that. So when we talk about quality, and we're basically talking about a lack of noise, more signal from the tissue, less noise from other things. But what is noise specifically? So this is a really nice table, which kind of shows the difference between where we have potential sources of noise, but what the neuroimaging community in large has decided whether they are actually going to get rid of it or not. And actually, quite neatly, NiReports tools tend to focus on calculating evidence of the upper two, which are the ones that are consensually classified as noise. So these include hardware interfaces, so ghosting, other artifacts, signal, susceptibility effects, but also physiological effects like breathing or respiration or head motion. So essentially, these things are things that we know will not be the signal of interest that we're trying to model, whereas the 2 columns in the bottom, maybe less so. Maybe we do want to actually look at that. So we're trying not to put any assumptions on the point of the researcher in that respect. So MRIQC is a tool that generates subject and group reports of quality metrics, because quality is highly subjective and the ratings are dependent on the experience of the rater. So we have regularly had some issues with trying to, teach new people how to spot bad quality images, and they get nervous because they're worried that they won't be able to spot it. And generally, it's definitely, there are some images that are obviously bad, but not all of them are. So MRIQC helps to overcome this by generating objective quality metrics that have been suggested throughout the neuroimaging literature. And these metrics provide criteria that allow people to make objective inclusion and exclusion decisions of their data quality before proceeding with any image processing. So it really, it saves the effort of having to process everything and then say, oh no, actually maybe that image didn't look so good, I'm just going to exclude it later. If you know that the data quality is bad from the offset, then you can actually save yourself and your results a lot of hassle in that respect. So MRIQC has become a very popular tool for human imaging quality control, but we have recently worked to provide some changes that allow it to work with rodents. So it still uses the same really simple command line interface with minimal user intervention. The only difference is that we added this new parameter flag with, it takes 2 arguments, either human or rat. If you leave it out, then it defaults to human, so it still runs the same way as it ever did. But if people include this -- species rat, then there are a number of changes that allows us to use the exact same tool as in humans as with rats. So things that change include that we use the Fisher 344 rat template that's downloaded from TemplateFlow instead of the MNI template that's usually downloaded. The BrainExtraction is changed out to the Arch BrainExtraction that we just discussed instead of the AFNI 3D skull strip workflow. And key parameters are adjusted based on the metadata from BIDs. These include things about voxel sizes and field of view that hugely make this workflow much more successful. This is an example of the workflow, and if anyone is familiar with MRIQC, hopefully you can recognize that these are exactly the same steps as the ones in the human workflow. They're just made with rat images instead, and we still get the same quality criteria, which allows us to make similar assumptions between both humans and rodents, that we can make criteria, principled criteria decisions, the same ones between both humans and rats. So lastly, I'd like to talk about fMRIPrep rodents, which is a preclinical pre-processing workflow specifically for functional MRI. It's based off of the success of fMRIPrep, which has become a staple of fMRI pre-processing because its minimal user intervention makes it very accessible, but also it's robust to individual data. So you can run the same command and it will be able to generate really robust results. But critically, the output is also really comprehensive in terms of the error reporting, but also it features these reports that we talked about earlier and the citation boilerplates that allow users to be completely transparent about both the quality of their output, but also the steps that have taken place to get them to this point. The outputs are also agnostic to the data analysis, which isn't necessarily true if you're doing one of these nose to tail pipelines where you submit your data at the start and there's an assumption made, maybe in terms of smoothing or in terms of regression of noise. None of these assumptions are made in fMRIPrep, allowing you to take whatever output you want and go on to focus on the interesting questions that you want to do with your data. The best part is that actually many of the key elements of fMRIPrep have already been covered, in the sense that You know, we've already talked about template flow and how just switching out the template can actually make a huge difference for whether we're using humans or rats, but also the skull stripping. So just by changing the skull stripping to a more generalizable skull stripping method, not based on shape or contrast, but instead based on atlas-based extraction, allows us to have a lot more flexibility in our workflow. Admittedly, there are some points in the workflow that are not applicable to rodents. And I mean, I think I've still to see a convincing argument about surface-based reconstruction for rodents. But if it comes up, we can come back. If someone has a convincing argument, I'm happy to listen. But as far as fMRIPrep rodents is concerned, we just skip these last steps. And some of this is unfortunately still a work in progress because it is a much larger workflow than than MRIQC, sorry. But with that, the last sort of big thing that's outstanding is the susceptibility distortion correction, which allows us to correct for the phase encoding distortions that we see with EPI images. And because this still works the same way in principle, we just need to make some adaptions to the underlying code in SDC flows to hopefully have a full workflow that is analogous between both humans and other species. So we're hoping that in the future, currently fMRIPrep rodents is its own separate package because of the speed and resources available to fMRIPrep. It's growing every day and improving every day. So in future, we'd hopefully merge fMRIPrep rodents into fMRIPrep like we did with MRIQC and the species flag, so that researchers can use the same tool with this, you know, becoming more and more ubiquitous in our processing, regardless of species. And we can have this access to the front line of the processing methods available regardless of your question. So yeah, if there's a take-home message, it's just that we shouldn't really assume that our data come from adult humans anymore. There are definitely enough tools out there that mean that we can provide flexible workflows that can really make it useful for anyone to use your software. Also, I'd really like to, regardless of whether you use, you know, preclinical data or human data or any other population, I'd like you to think about the assumptions underlying your data processing and to challenge them, because the case of preclinical imaging with human data processing software is a stark example, because these two images look very different and have a lot of differences between them. But it doesn't, it's still a point to be made that what if you're studying a population that is less different than, you know, humans and rats, but it's still different all the same? Maybe the assumptions that are made for yours are maybe not as valid as you thought. So although changing defaults is prone to researcher bias, there are tools out there that allows leverage of the metadata that can be available, and also gives can have other tools to underpin the workflow that provide us with flexibility to change our parameters based on the data that's input and not based on what works with the average adult human brain. So finally, I want to reiterate that I don't think that this problem will be solved by a single lab coming up with and distributing their approach. I think that it will only really work if we make this an inclusive community effort. So with that, I'd like to say, if you're interested in any of the projects, maybe you are preclinical imaging but not small rodents, you know, but anyone really, if you'd like to contribute to these projects, please feel free to get in touch with any of the projects I've talked about today. So with that, I'd like to thank you for your attention. And yeah, I have thank all these people, my group at the Brain Centre, who let me, you know, play with all of the data. and the NiReports community, especially Oscar, and for Russ, who invited me to come over and start working on this, and MRC, who allowed me to go there. So yeah, thank you very much, and I will take questions.
Thank you, Eilidh. That was a really wonderful talk. I don't know, I'll be honest, I don't know much about preclinical imaging. I work on baby brains, but it was really interesting to see how some of the challenges that we encounter in sort of fetal and neonatal research are actually quite similar to the ones that you were talking about. So yeah, thank you for that.
No problem.
We have a few questions, so I'm gonna go through them in the order sort of how upvoted they've been. So we have a question from Anonymous saying, excellent work. To be a devil's advocate, it seems that both the preclinical and clinical imagers have found solutions to their problems, albeit divergent. What do you think would be the benefit to human clinical imagers to make their tools easy to be uptaken by preclinical imagers?
Yeah, no, that is a good question. What do you think would be the benefit to make their tools easy to be uptaken? So I think the benefit is the sustainability of their approach. So at the moment, you know, those pipelines that I showed before, they're mostly driven efforts by a single group. But quite often, you know, as we've found in a lot of the reproducibility issues, is that a lot of code might become less useful when a particular postdoc or PhD student leaves. The NiReports community has a lot of resources behind it that allows it to, it has software developers as staff that allow the tools to actually be developed. They can find bugs, they can address questions about users, and they can you know, work with the community to include new features as the field progresses. But one person trying to keep up with the entire field of neuroimaging is just, you know, not feasible. It's not sustainable. So the benefit is really for both for human and clinical imagers to, you know, be able to translate their work. They can actually get to use, you know, the cutting line efforts in data. If they can get their hands on some preclinical data, they can use the exact same analysis methods as they've been using in humans, but in rodent data to maybe test those assumptions. So you want to, like, the thing about it is that the reason we do preclinical imaging is because we can't do every method under the sun in humans, and without preclinical data, they might not ever be able to really pin down those biological underpinnings in the way that they want to. So yeah, I guess that's my answer, is human and clinical imagers should be interested because you need the scale. You need to be able to sort it on different scales, not just on a macro scale.
Brilliant, thank you. That was such a comprehensive answer. Thank you so much for that. Okay, so the next one is, could you please comment on sample sizes in preclinical imaging? Those are, of course, very difficult studies to run with ethical considerations limiting sample sizes. Would combining data from multiple sites help increase your number? What are some considerations when doing this?
Yes, absolutely. I think that it is definitely something that we should be looking into, is to be more proactive with sharing our data. But there are certainly some issues around the fact that there's much more different flavors of preclinical imaging than of humans. So generally, when we are using preclinical experimental models of animals, we're looking after a certain question. So there's like knockouts, and it's which animal do you use? Do you use rats? Do you use mice? Do you use non-human primates? Do you use animals that have been genetically modified? Do you use, and then even within like rats, you might have like, these are sprag dollies, these are fisher, these are, you know, so there's actually a whole lot of variation within the field that is probably down much more of a question for the researcher at hand that probably precludes the sharing of data in that sense. I think that it's maybe a sense of like, why would I share this? People might not find it interesting, but I would say to those people, just share it anyway, because you don't know what other people are trying to do. But I do agree, if we combine data from multiple sites, I'm sure that we'd probably get there faster.
Thank you. That was a great answer. Okay, so we have a short question from Mo asking, have you also explored adapting the MRIQC to NHPs?
Yes, we have. So for other people who don't know, NHPs are non-human primates, and I get much like the rats, there's different scales of like which non-human primates. We have macaque templates, and I think we have another non-human primate template on MRIQC, but it's mostly just down to time where it's mostly me and a few others that are driving the preclinical imaging side. So if more people want to get involved, we would absolutely be able to achieve more. It is hugely down to time. It's something that we definitely want to move into.
Lovely. And I think we have one more question. So that is, what field strength do you typically use for your scans? Would the processing pipeline differ based on field strengths?
Yes, so at the Brain Center, we have a 9.4 Tesla scanner, which seems to be, well, I showed that range and it's sort of in the middle a little bit. I do know people who are using 7 Tesla scanners with maybe more powerful head coils, like cryo coils and stuff. So it's possible that your processing pipeline might change, but I would say that the fundamentals are probably, pretty solid. You just might want to, if you have a higher field strength, you have to be aware that there are probably more artifacts or particular artifacts like the susceptibility distortion correction. So it's probably even more of an imperative at these high field strengths that you have an accurate B0 field or that you can actually correct for. So although it might not be necessary in the lower field strengths, or it's less necessary, I don't think it may be a best practice processing pipeline should probably correct for it anyway.
Lovely, perfect. Thank you so much. So that's it with the questions, and that's actually great because we've just reached the hour. So I want to thank you again, Eilidh. That's been a really, really wonderful talk. For everyone anyone who's still here, thank you so much for attending. You can share, we're going to put the link to Eilidh's talk online, so you can share that with your friends and colleagues who weren't able to be here today. And if anyone still has, time off in their calendars today, do join the conference that's going on today, the Teaching and Mentoring Open Research one. It's going on until 5 British Summer Time. So you can check the website and Twitter for the link. And yeah, thanks again, everyone, and we'll see you soon.